So I'd like to start with thanking the IMP and the Birnstiel Foundation for this amazing recognition of my work. This is an amazing opportunity to visit here. So I'll focus on enzymes, and enzymes are these great green and efficient catalysts useful in many different industries. And for example, my model system for this project was zyanases, which can break down the xylene sugar chain found in plants and has a lot of application, for example, in biofuel production. Now, different proteins have different structures which dictates their function. So this is basic biochemistry. And those structures actually dictate their function quite precisely. So, for example, enzymes are perfectly adapted to process their substrate into products. So this means that if we want to design new enzymes, we have to control and reliably design their structure in order to get novel functionalities, especially the loop regions, which are specifically determining the functionality and specificity of proteins. However, if we think about the number of possible alterations to protein structure, so in mutations, insertions, deletions, it would be an astronomically large number, and which makes them very hard to predict. So despite years of research, there is no reliable way to design loop regions and structures of proteins. So in this project, we aim to design millions of enzymes, so we have different in sequence and structure, so we can learn statistically what governs high-efficiency catalysis in design enzymes, and how can we use this knowledge in order to improve our design method so we can design enzymes with new functionalities. Now, unfortunately, there is no practical way to synthesize DNA molecules encoding 1 million different genes. And I have to mention, we didn't want to use like random libraries in this project. We wanted everything to be designed by computer and rationally, so we can then understand what happened, why something is working or not. So it has to be designed. So instead, we switched to slightly different questions. So how can we design a huge repertoire of proteins from prefabricated protein fragments? Now, the question of how to construct something useful from pre-existing parts is actually quite common in engineering, so from construction to electronics. However, for this to work, the parts have to be modular in a sense, so they have to be compatible with each other. But one of the very basic observations about proteins is that they make a lot of a long-range interaction across the structure, so they're not modular. So this means that the vast majority of randomly combined protein fragments would not assemble into a folded protein. However, nature give us one example of a truly modular protein fold. This would be obviously antibodies, which is by far the most versatile protein binder system that we know of. So upon infection, our bodies can generate billions of different antibodies just by recombining a few subdomain gene fragments, as shown here. So basically, this shows that a limited set of genetically encoded domains can actually generate a limitless diversity in proteins. Unfortunately, there is no parallel mechanism for diversifying enzymes in such way, nothing that comes any close to that. So here we wanted to ask, can we design a set of protein fragments that when we let them to freely combine in a test tube, would assemble into folded and active enzymes? And in this project, I focused on the Timber fold, as you can see here, and it shows it because it's known to evolve modularly, and also it hosts a lot of enzymatic reactions in nature, so it will be useful for many different design applications. But you have to be very careful when you design such a protein from fragments, because as you can see here, the active site residues are actually located all over the protein. And when you design an active site, you have to be extremely careful, because if just one atom is not in the right place, you won't get any catalytic activity. Okay, so how does this work? We start by collecting all the available structures for protein family, in this case, the zyanases, and we segment them along structurally conserved regions in the protein. So here I segmented them into 4 linear regions. So now I don't use the natural sequences for this fragment. Instead, we introduce a lot of mutations in order to make them more stable and also increase active site diversities, which might lead later to different specificities and functionalities. So if I design roughly 30 fragments for each of the 4 segments, so 30 by the power of 4 gets me to roughly 1 million designed enzymes. We continue and order DNA molecule encoding different gene fragments, and we let them to clone all against stone in the test tube, and then clone them into yeast for a software display. And then once the proteins are displayed in yeast, we can actually label the ones that are active, and we do that using an activity-based probe that specifically labels the active site of enzymes that can break up this sugar chain. And once the proteins are labeled and in yeast, we can use a fax machine to sort the active population and the sequence this population and go back to the computer and try to analyze what differentiates the active and inactive enzyme in those proteins, so we can learn what governs catalysis in those proteins. And if we learn something useful, we can embed it into the pipeline. And if it is useful, we should get a higher success rate in the second library. So this is exactly what we did. And after the first round of this design, we got thousands of different enzymes that were active, adopting hundreds of different structures. So we wanted to test a few structural and energy matrix that discriminate between the active and inactive enzymes. I have to mention, we didn't use any features that are specific to xylanases or the sequences themselves. We wanted matrix that are general and useful to different enzymatic systems. We didn't want just to improve for this family. We wanted general stuff. So for example, these two matrix gag the core backing of the protein. So for example, you can see this green fragment here. So it was selected for the first library, but according to these two matrix, it's actually signaled as a bad one. You can see why. So here, this white region is actually an unpacked region of the protein. So obviously, this would lead to very high energies, and this protein would never fold. Other metrics were, for example, the overall energy of the protein and the hydrogen bond energy of the proteins. So for example, this red fragment was again selected for this first library, but signaled as a bad one according to this metric, and you can see why. So during design, we mutated these two key arginines that made a lot of hydrogen bonds in this loop, but obviously when you mutate those, the loop would not fold correctly and this enzyme would not be active. So these are some of the matrix that we use, and you can see that while they're all statistically can differentiate between the active and inactive proteins, they're not good on their own to have a good discrimination. So instead, we combine those into a logistic regression model, and now we get a very nice separation between the populations. So we call that the activity predictor. And to test whether the matrix that we embedded in this activity predictor are actually predicting activity, we designed a second library, and you can see that, at least on the computer, scores look very good. So we went on and tested it in the lab, and very encouragingly, we got an order of magnitude increasement in success rate compared to the first library. So this time we didn't use only the first activity-based probe that mimics the native substrate for this protein family. We used another one that mimics a different sugar chain. And you can see that we got quite a lot of hits here as well, meaning that our library also was able to show some specificity changes in the active site. And the proteins in the library are very diverse, so they adopt very different structures and also a lot of diversity in the architecture of the active site itself. So again, pointing that we might get different functionalities. Also, we got a lot of diversity, so up to 140 mutations away from any natural protein, including a lot of insertions and deletions. The proteins are very diverse in length. And also, most of the incorporated protein fragments come from 4 different sources, which would be quite challenging doing an hybrid combination. And I also have to mention that 75% of the active proteins in the second library actually incorporated a fragment that was not present in the first one, meaning that the activity predictor was able to generalize to things that it didn't see in the first, in the training data set, which is quite challenging in machine learning. So to generalize to things we didn't see, and we think it's because we use those general matrix that are general to all enzymes. And we also wanted to test whether the activity predictor can be also useful in a different design task. So instead of designing a huge modular library, we designed a small set of proteins, and 25 out of 27 that we tested were actually active. And you can see some of them here having catalytic activities comparable to xylanases coming from therophiles. And just to mention, xylanases evolved for years in cow guts, so they're highly active, they're super efficient, and our design are comparable to those. So that was very, very exciting for enzyme design. So just to wrap up, I show you that when selecting correctly protein fragment data module, you can generate a huge diversity in structure and sequences. By analyzing this huge data set of designed enzymes, we showed statistically and robustly for the first time that active enzymes rely on stable and compact proteins that are pre-organized in the active site for catalysis. Also, I showed you that by designing rationally libraries, we can actually learn from the experiment and to improve our methods. And lastly, again, because we designed those using understanding of how the structures of protein work, we managed to get substantially more active enzymes than random processes. And with that, I'd like to thank my mentor, Soraya Fleischman, and my colleagues, especially Olga, and our great collaborators from the Leiden University for the activity-based program, and the Rothschild Foundation for funding my PhD. And thank you.